Good day once again. Welcome to Harris Impact International Academy, and today we are in our last series. We're in the last session of this series where we're going to be describing fungi reproduction. We've talked about bacterial reproduction. We've looked at viral reproduction. So now, reproduction in fungi. Let's quickly dive right in. You see, fungi can reproduce in two major ways. We have the asexual mode of reproduction, and we also have the sexual mode of reproduction. So we have asexual mode of reproduction in fungi, and then we have the sexual mode of reproduction. Very good for you to know that the most commonest form of reproduction in your fungi are the asexual mode of reproduction, basically by spore formation. By spore formation, what you can call sporulation. By spore formation. Yes. While your sexual mode of reproduction is basically by fusion of genetic materials. Yes. So there is fusion or exchange of genetic materials in this case here. So what really happens during this asexual mode is that we have a spore as usual. That spore is being produced from what we call the mycelium. So just imagine that you had the mycelium here now. Let's take for instance, we have the mycelium. You have the mycelium. This mycelium, these are the important points you want to take note of there. The mycelium is usually having genetic number of n. So it means that it is haploid. It means that it is haploid. So you have an haploid mycelium. You have an haploid mycelium. Yes, this haploid mycelium will utilize mitosis to produce a spore, meaning that the genetic number of that spore will also be n, haploid spore. Very good. So you are having from here now, you are going to be having spores. Idea now, spores that will be produced within spore keeping, or you can say spore storing structures, right? What are these spore storing structures we're talking about? We're talking about a sporangium or a conidium. So it can either be a sporangium, you can either have it as a sporangium, or you call it a conidium. So the sporangium there. So this is from the mycelium. There will be growth of something we call a sporangiophore. The mycelium is like a root-like structure that's made up of hyphae, collection of fungi hyphae. So from that mycelium, you have the growth of a sporangiophore. At the top of the sporangiophore, you see the sporangium, which is going to be a sac that will be holding the spore. So it's within that side there that these produced spores will mature. So when they get matured, they will now get released. So from here now, we are now having what? The release of matured spore. Matured spore. Now when these matured spores are released, they get dispersed either through air, water, wind, animal, or anything that the case may be, explosive mechanism and all. So here now we are having the release of mature spore. Then the final thing that we'll be having is the dispersal. We have the dispersal. And I told you that this dispersal can be via wind, which is air, can be through water, it can be through mechanical means. So once they've been dispersed, are you getting me now? They will now grow new myceliums. So from the new mycelium, a new fungi will grow. So that's how your fungi reproduces asexually. So this is an asexual mode of reproduction. Now, if we're talking about the sexual mode of reproduction, it's very, very simple. The sexual mode of reproduction has something very interesting. Now, you have two kinds of mycelium. You have the positive mycelium, you also have the negative mycelium. Just the way you have the bacteria, where you have the F positive and the F negative. So in this case here, just imagine that you had this here like this. I'm just picking a single hyphae from the mycelium. We have this hyphae here, we have this other hyphae here. Let's imagine that they have crossroads for instance. Let me just assume that. So let's just take that for instance. This is not spirogyra, please. It's not spirogyra. Not spirogyra. This is the crossroads. So let's assume this is the positive and this is the negative. Positive are usually in bacteria. Positive are usually the donors. Negative are usually the what? The acceptors. But in this case here now, the positive is going to be standing more like the male, and this one is standing more like the female. So good. What happens here is that these two fungi relay side by side and later on come in contact with themselves through chemotaxism. Again, now that chemotaxism is as a result of enzymes that are produced by the negative mycelium that will attract the hyphae of the positive mycelium closer to itself. So once that happens, I'm not saying that there is going to be actual motility. You understand? That's why we did not say we're talking about contact from when they are regional contact. So there is going to be regional contact, and then when they come in contact with themselves, first thing that will happen is this: you'll be having something that we call plasmogamy, which was asked last 2 years JUPEB. Plasmogamy. So remember that the mycelium is N, right? It's N. Still the same thing in this place, N. So in plasmogamy, what's happening is that mycelium hyphae that are in close contact will fuse. That their cytoplasm fuses. So the myceliums become in contact and then they will fuse. And when we say they will fuse, that means there will be disintegration of the central piece of this of these two mycelium. So you'll be having something that will now look more like this. Let's say from these two here now, you'll be having them looking more like this. That's at this region when they will fuse, you'll see them looking like this. Just have now a single large conjoint guy. So you see it now. So before it would have been like separated here, but now they fused. Now that they fused, let me show you something that was happening initially. Initially, this guy had one nucleus. This one had one nucleus. But what's going to be happening now? We are going to be having a state where you're having this guy, this is one nucleus, this is the other guy, one nucleus. So the nucleus did not fuse, it's only the cytoplasm that fused. That's plasmogamy, right? Now, it means that plasmogamy will now lead to a state in the life cycle that we call what? A state that we call heterokaryotic stage. So the heterokaryotic stage now simply means what? A stage where you are now having two unfused nucleus within the same cytoplasm. You get what I'm saying now? So you're having two unfused nucleus. There are two unfused nucleus within the what? Within the cytoplasm. We know that this cytoplasm now is fused. It's fused cytoplasm now, and that's the reason for the two nucleus. Very, very important. So it is in fungi that you see heterokaryotic stages or heterokaryogamy. So when you hear heterokaryogamy, is the presence of two nucleus as a result of plasmogamy. Now, when this heterokaryotic stage occurs, the next thing that you want to take note of there is that they can ask you during the heterokaryotic stage, what will be the gene or the chromosomal number? Now, don't be tempted, because now we know that in plasmogamy, I told you mycelium is N, so we have N here, so this is like N here, and N. So now, we are now having N and N. Don't be tempted to say 2N, they didn't fuse. So the genetic number of the heterokaryotic stage is still N, it's just that you're having N plus N, and it will not be equal to 2N during the heterokaryotic stage. So what will happen there is that this guy here will move from heterokaryotic stage into what we call karyogamy. So you will now have karyogamy. So the two major steps in your sexual reproduction in fungi is that you have a stage you call plasmogamy, and then you also have a stage you call karyogamy. Plasmogamy leads to heterokaryotic stage, karyogamy leads to homokaryotic stage, where the two genetic numbers will now fuse. So in karyogamy, what's happening there is that what? Is that the two nucleus now fuses, forming diploid chromosome. So here now you can now have the diploid chromosome. Very, very simple. So in this place here, chromosomal number is 2N. In this place here, chromosomal number is what? N. Very, very, very important. So after this happens, what happens there is that this guy here will now look somewhat like this. It will look somewhat like this. So you just have a single nucleus now. So this is where the fusion happened. So that's what's happening in that place. So what kind of chromosomal situation are we having here now? Or nuclear situation are we having here now? We are now having the normal homokaryotic stage. So good. When this happens, you will now have the meiosis occurring. Very, very important. What happened in this place was mitosis. I said it, mitosis. So in this place now, you will now see meiosis. And if meiosis is happening to this 2n, remember that it is 2n here. Meiosis will lead to the formation of what? Of chromosomal number what? N. Remember meiosis, you have 2n turning into what? You have diploid chromosome turning into 4 haploid chromosomes. So you're having your what? 4 haploid chromosome here. So that's what we're having. So it is this N, that's this N stage of this fungi life cycle that will develop back into the what? Mycelium. You can see the mycelium that is N. So this describes your sexual life cycle of your fungi. It's very, very, very easy. So basically, one thing I just wanted to take note of here is that during this meiotic side, when you're moving from homokaryotic stage to this part where you're producing this N here are going to be in their spore stage. It's going to be in spore stage. So later on, the spore will start producing these myceliums. So it's going to be like in the spore stage. So this kind of spores are the ones that are calling sexual spores. Where you be hearing spores like your Basidiomycota, that's sexual spores. You have spores like your Ascomycota. Unlike this one where we said that the storage spores keeping structures as Brangiosperm and Conidiospore. In this case here, you have your what? Your Basidium. In this guy here, you have your what? Your Ascom. Some people will call it Ascos. So something like that. You have your Zygosporangiosperm as well. That's the one for your Zygomycota. That lecture would have been taught in the previous lecture. If you have not watched that video, you go back to the previous video. You will see where these things were referenced. So what I'm just trying to tell you there is that is what brings the differences between sexual spores and asexual spores. So the asexual spores are the ones you have here, produced directly from the mycelium, mitosis, and the sexual spores are the ones that were formed from the homokaryotic stage, meiosis, and produce those haploid spores again. So that is that for fungi reproduction. So I believe that the lecture would have been very helpful, and if it was, do well to comment in the comment section downwards or down there, as well as subscribe to our channel. Also click on the notification bell so that whenever we drop more content like this, you will be notified.